Hi everyone, this is Professor Winans, and this is our very first video for Biology 389 Metabolic Pathways. What I've done here, what I'm going to be doing with the videos this semester, is using my document camera that I have here. I'm just going to be writing notes. I've printed out the PowerPoint slides so that I can write notes on them. In this case, the notes are mostly pre-written on the slides, so we'll just have them as we go through. In other cases, I will be writing extra notes on a separate piece of paper, which might happen in this video as well. But just so that you're aware, these are the actual PowerPoint slides called Unit 1, and we will go through them slide by slide so that you can follow along with this video while reading the slides. If you want to toggle back and forth, pause the video, look at the slide, read over the slide. However you want to use those slides, they are presented here, but you have them also posted as a PowerPoint in Blackboard. So what we're going to talk about with this first video is an introduction to what metabolic pathways are. You're already familiar with some metabolic pathways. You've learned about the Krebs cycle, for example, starting with glycolysis, going into the Krebs cycle and the electron transport chain. You've already heard of those things. We're going to be looking at those in more detail and looking at different pathways, different branches, what are called shunts. So if a reaction goes in one direction rather than another direction, sort of like a side pathway, that would be called a shunt. So we'll be looking at those sorts of things, and then how all the pathways interact with each other, how they're dependent upon each other, and how everything relates to the organism or the person, the entire body as a whole. So we're going to be seeing in this particular video just the introductory kind of material. We're not going to be digging deeply into specific metabolic pathways here. You will see some very specific metabolic pathways, but we're not going to delve too deeply into them on this first day. So here we go. What is cellular metabolism? It really is just referring to the various chemical reactions that can happen inside of a cell. So examples would be glycolysis, the Krebs cycle, things like that you've seen before. But there's many, many other reactions that happen within a cell. In A&P 1, you probably would have learned about second messengers. Those are chemical reactions, and they use enzymes, and they have several steps in the reaction, and that is typical for any sort of metabolic pathway where you have certain substrates, where a substrate is something that's being worked on. It's a molecule that's being acted on by an enzyme, and then you get an intermediary product, which then leads you to maybe another reaction and then another intermediary product. So for example, let's say we have this substrate, whatever that substrate might be, and we're going to have a reaction here, and there's an enzyme, whatever this enzyme is. And normally the enzyme is, ends with a ASE in the name, so it makes it easy to remember an enzyme, or to know or recognize that that is an enzyme. So we have our initial thing here that's being worked on. We have the enzyme that works on it, and we get our product. And this could be the intermediary product, because we don't typically just have a one and done sort of chemical reaction. We tend to have more chemical reactions. So let's put another enzyme here. Put ACE at the end. And then we have another product. This product is probably an intermediary product too. There's probably many more steps to this, but you get the idea. We'll just have this be our final product, and we have a very short metabolic pathway right here. Now, this metabolic pathway, pretty simple, requires two enzymes here. If we were missing either of these two enzymes, then we're not going to get to our final product, right? So both of these enzymes are required. So the ability to make both products are required just to get to that final product. So if you're missing any of them, you're not going to be able to do this pathway. Or if any of them have been inhibited for various reasons, which we'll see different reasons why some enzymes might be inhibited as we go throughout this semester, then, if you inhibit this enzyme, you're not going to go down this pathway. Additionally, there can be other competing enzymes that might be taking this in a different direction. So this could be a shunt. So here's our different enzyme, and we have a different intermediary product. And then that would take its own pathway. We'd have another enzyme over here and another product. So something completely different, just this whole different chemical pathway, just because we had maybe more of this enzyme, or maybe this one was inhibited and this one was stimulated, or this one was just more competitive or more active. So we would call this a shunt because it's a separate pathway off of the initial main pathway. And we're going to see lots of examples of these main pathways with little side pathways or shunts going on over here. But you can see already how complex this can be with these chemical reactions happening, all dependent on each other and all kind of relying on each other and moderating each other in a way. Maybe a little more of this, a little less of this, or vice versa. And it's all to keep a nice balance inside the cell. All right, so looking at an old familiar chemical reaction, glycolysis. Glycolysis has probably been presented to you just on its surface. It's something that happens in the cytoplasm. It's anaerobic, so it doesn't use oxygen, and it gives you 2 pyruvic acids, or pyruvate we would call it, so 2 pyruvates, and 2 ATP. So we get a little bit of energy out of glycolysis, but we're really not getting a lot of energy out of that. What we're really looking for here, what's going to give us the most payout in the end, would be the pyruvates. Now all of this glycolysis, the word glycolysis literally means breaking down glucose. So glyco referring to glucose, or glycogen, but in this case it's talking about glucose. Lysis means to break down, so glucose, we're breaking it down. And this is, so just taking, your glucose, turning it into pyruvic acid or pyruvate and 2 ATP, this is glycolysis in a nutshell. But there's actually several steps to it, different phases. One phase is called the preparatory phase. And the other would be called the harvest, or you could call it the payoff phase. And we'll see this several times. We're going to look at glycolysis a little bit today. I'll show you glycolysis, but then we'll also really delve into it or look at it more solidly in the coming weeks. So there's just a bunch more steps than what has been presented in the past. There really is a lot more to it. Lots of enzymes, lots of intermediary products. So here is the metabolic pathway for glycolysis. Oops, that's not working. There. Okay, so this is a very small slide. I don't expect you to be able to see what we have written here, so I have actually written it out on a different piece of paper. Still hard to see, we can't see the whole thing all at once, that's okay. It is on a slide and that you can zoom in on and look at more closely, but I do want to let you know before you go, oh gosh, what are we going to do? Why do we have to look at all these terrible chemical names? for a test, you do not need to memorize these chemical pathways. You do not have to memorize all of these names here. What you do need to be able to do is to look at an image like this. So just the chemical pathway, not what I've written all around it, but just have an image presented to you as part of the test and be able to interpret it, be able to read it, understand what's going on. And that's what we're going to be working on. So you will be able to do that looking at that image. But don't feel that you have to memorize all that. I don't think anybody could memorize all of that. It's just not something that's worthwhile doing anyway, when you can just look it up and see it. So no memorizing, just understanding here. So what I've done here is put the names of the different products. So we start out with glucose, so that would be the initial substrate, and then we go through a bunch of chemical reactions, and everywhere in green would be a product that's being formed with each chemical reaction all through the 10 steps. So we start out with number 1, which is turning glucose into glucose 6-phosphate, and we go all the way down to step number 10, and the end result of step number 10 is 2 pyruvates. Over here in the purple column, we have the enzymes. So, for example, in step 1, we have 1-hexokinase. That's an enzyme that helps to turn glucose into glucose 6-phosphate using ATP. So here, when we have, not every step has these little pink sides, but some of the steps have these little pink kind of extra things going on with the reaction. This is where something is used or something comes out of the reaction that is not part of the main product and is not part of the enzyme. But here, this reaction requires ATP. We're breaking down ATP, and you end up with ADP. So this is actually using energy. We'll see down here that we're, again, using energy. We're taking ATP, we're breaking it down into ADP. And you think, okay, well, glycolysis is supposed to be about making energy. And it will be. This is the preparatory phase. We are preparing to start making energy. We've used 2 ATP, but we're going to get a net of 2 ATP out of this, so we're going to end up making 4 more ATP, if it makes sense. So if you use 2 ATP and then you make 4 ATP total, so it took you 2 ATP to make 4, so your net is 2 ATP. Okay, so all this is about preparing essentially to net that ATP. In here we have another little extra thing where we're adding phosphate into our molecule, or adding another phosphate, I should say, into our molecule just by using a phosphate alone, plus 2 NAD. NAD probably sounds familiar to you from when you've heard about the Krebs cycle in the form of NADH, which there's some more NADH. So we're going to see more NADH as we go through the Krebs cycle ourselves and looking at what this NADH that came out of glycolysis, what's going to happen with that, where we take it, etc., etc. We will look at that eventually, not today. But in a future video we'll look at that. So just kind of keep that in the back of your head that we're getting some, we're taking NAD and converting it into NADH while we're kind of incorporating more phosphate into our main products system over here. Okay, now we can start harvesting some ATP. So we're going to take ADP, so that's the form with just 2 phosphates. We are going to add a phosphate to it to make ATP, 3 phosphates on it. And there's 2 energy. We need 2 more, of course, so here we do it again. Oops, I should have a 2 on here. 2. Happens twice. So 2 ADP, and we get 2 more ATP. That's our total of 4, which it cost us 2 to get in the end. Okay, so essentially, there's a couple of things that we want to take out of this slide. which we'll go over to here to show you that. Here, we need to know that these are the products. You can always recognize the products just because of their names, like they just are, biphosphate, glycerate, all these eights at the end. That is a product. You're going to see a lot of, there's other products and other pathways that all seem very recognizable as products. If you see something, whatever phosphate, whatever glycerate, whatever pyruvate, that's going to be one of the products. And over here for the enzymes, look at them. They all have an ACE at the end. Hexokinase, isomerase, fructokinase, aldolase, that's an easy one. So they're always going to have that ACE at the end, so you know that that's an enzyme. Also, to make life a little bit easier, they're very much named after what product they're working on a lot of the time. So phosphoglucose isomerase is working on this molecule here, which could also be called phosphoglucose. It's glucose 6-phosphate. It's a form of phosphoglucose. Phosphofructinase, fructose 6-phosphate. So you can see how those are named similarly. This one, unfortunately, breaks that pattern, so it doesn't, quite go together. It has a different reason for why it's named this, where sometimes enzymes are used in different pathways as well. And that actually pulls me to my next point, which is what we do with the pyruvates after we're done with glycolysis is that they're going to go into the Krebs cycle. But it's not just magic. They're not just going to jump into the Krebs cycle. They need to first go into the mitochondria. They need to also be converted into acetyl-CoA, because acetyl-CoA is the molecule that can enter the Krebs cycle. So that is one thing I do want you to just remember. One not so hard thing to remember, acetyl-CoA goes into the Krebs cycle. This is going to be true whether we're starting with glucose like we just did with glycolysis, or whether we're starting with a fat or a protein. We're always going to be trying to get acetyl-CoA if we want to go into the Krebs cycle. You just got to get acetyl-CoA. Now, the way that you turn pyruvate into acetyl-CoA is through use of an enzyme called pyruvate dehydrogenase. It knocks a hydrogen in the water, or a hydrogen and oxygen, I should say, hydrogens and oxygen, essentially a water. It takes that off of pyruvate, dehydrates the pyruvate essentially. So pyruvate dehydrogenase is an enzyme that we're going to see here, taking our pyruvates, turning them into acetyl-CoA, so we can go into the Krebs cycle. And we're going to just keep seeing pyruvate dehydrogenase kind of over and over again in terms of whether we're kind of stimulating it or inhibiting it. If we're stimulating it, then we're making it easier to get energy because we're making it easier to get acetyl-CoA. If we're inhibiting it, then we're not going to make acetyl-CoA so easily from our pyruvate. We're not going to do the Krebs cycle so well, and so we won't get so much energy. So we're going to be seeing acetyl-CoA a bunch and pyruvate dehydrogenase a bunch. So that's one point, that enzymes can be used multiple times in various pathways. They can be inhibited, they can be stimulated, and we're going to see all that as we go through this semester. Now, another thing that we need to think about are just the words proximal and distal. These probably sound very familiar to you from A&P 1 and 2. It should be part of your vocabulary. If you've forgotten, here's a quick refresher. If something is proximal, it's closer, and if something is distal, it's farther away. So when we think of it in something like anatomy terms, it would be like if you're looking at, let's just use my hand because it's here. So obviously my shoulder would be up that way. So here's my wrist, here's my thumb. So this would be the distal end of my thumb because it's farther away from my shoulder, which is the attachment to the body. And then as we get closer to the wrist, that would be more proximal. and we would keep moving proximally all the way up to the shoulder. When we apply it, though, to a metabolic pathway, it's a little bit different because, you know, there's no attachment part, but we're just talking about the beginning versus the end. So if you think of proximal as the start of the chemical reaction and distal as the end of the chemical reaction, then it makes sense. So proximal, like the most proximal thing would be the glucose. The most distal thing here would be the pyruvate. Now, on a test, how we would talk about that, or how I would use that, is you would get your, you know, your glycolysis metabolic map. So you would see this thing here, but it would be visible instead of tiny like this. You would see it, and I might ask a question. Let's just do it from the top here. Like, let's say we inhibit this enzyme. So you'll see all the enzymes, you'll see all the products here, and I'll say we inhibit this enzyme. What would the effects be on this reaction? Well, there would be no effects proximally. So if you inhibit aldolase, we have no effects on steps 1 through 3, but distally we do have effects. We've inhibited this enzyme, so that's going to slow everything down from there until the end. So we slow down steps 4 through 10, or stop them entirely, depending on how strongly we've inhibited the aldolase. So that's the explanation for it, but I might, you know, you might have it worded as something like, instead of just saying we've inhibited steps 1 through 10, we inhibit proximal to step 4. So that would be steps 1, 2, 3. And, no effect, no effect, proximal to step 4. And we inhibit distal to step 4. So no effects proximally, no effects before, yes effects, inhibition in this case, distally. So after that. So if you want to think of it as just before and after, you could think of it like that. Or I might say, I might kind of flip it around and say something like, we inhibit the enzymes proximal to, let's just pick the same spot, fructose 1,6-biphosphate. So essentially you're inhibiting all of those enzymes up here. So those would be the choices of the enzymes that got inhibited. So in that case, since I inhibited us right from the start, the whole thing is inhibited. If I inhibited the enzymes distal to step 4, then we're inhibiting all of these. We have no effect up here. We only have our effects down here. So just be able to interpret, if I use the words proximal or distal, be able to find that. Or it might be even more simple. It might be just something like, what is the enzyme that is proximal to step 5? So you find step 5, you look proximal to it. Or what is the product that is proximal to step 5? You would just look to the one right before. What is the product that is distal to step 5? And you would just look right there. So that's how those would be used. Okay, now before I flash that at you, I do want to remind you that you do not have to memorize these. And the one that I'm about to show you is kind of an ugly one. It's a very complicated one, but there's a whole point to why I'm showing it to you, which is just to show you relationships between these metabolic pathways. So we see this one. in all its glorious complexity of just glycolysis. But just imagine if we were looking at not just glycolysis, but the Krebs cycle, and not just the Krebs cycle, but using fats or proteins in the Krebs cycle, or making fats and proteins out of Krebs cycle intermediaries, or making glycogen out of glucose, and we end up with something that looks like this thing right here. So this is too tiny to really see at all. Even on the slide, you'd have to zoom way, way in. So I don't want you to sit there and look at all the different things, all the little writing on there, and all the little arrows going every which way and drive yourself crazy. What I have done is just put in big magic marker what the main points are, what the main thing is going on. So let me explain what I did here. So here in the middle we have glucose. So glucose, this is our glycolysis that we just saw. So right here, that's this. Squished really teeny, teeny, tiny. And then we go into the Krebs cycle. So we get acetyl-CoA, so down here, pyruvate with pyruvate dehydrogenase. Gives us acetyl-CoA. And acetyl-CoA goes to the Krebs cycle. And so that's what we see here. And that's just kind of happening in the middle of this particular metabolic map that we have. Now we can just get energy, so yay, that's like, glycolysis, Krebs cycle, energy, done. It could be like that. But we could also go onto one of those shunts, do a side pathway. So let's take our glucose and let's say we're going to do storage of extra glucose. So we're going to make glycogen. We can take that into the glycogenesis pathway, which we're going to look at later on, not today, another day. But we'll see glycogenesis going off of the main glycolysis pathway. We'll see something called the pentose phosphate pathway. This is one of those shunts. It's only one. So it's a shunt off of glycolysis. It happens when you have more of like a high energy state. You already have a lot of ATP. You can end up doing this pentose phosphate pathway instead. So there's, this is something that we're going to look at a little bit. If we go off of the Krebs cycle itself, we can incorporate proteins. So maybe proteins are being made out of this. So you can make quite a few different types of proteins or different types of amino acids inside your body. You can't make every amino acid, but you can make a lot of them. So that can be happening. Or those proteins can be being used for energy themselves, broken down. It's not a great idea to do that. You don't want to do that very often, or too much anyway, but it does happen. So we can, break them into acetyl-CoA, and there you have the Krebs cycle, going into the Krebs cycle. Same thing with lipids. We can make lipids, fatty acid synthesis, which we're going to be looking at for our unit 2. Proteins will be unit 3, fatty acids and lipids are going to be unit 2. But we can be making them, we can be breaking them down and putting them into the Krebs cycle for energy, which is something that we do all the time, and that is, a very efficient source of energy. And all of these pathways rely on each other, just depending on each other for regulation, essentially. So are we making a lot of energy from lipids? We don't really need to make a lot of energy from glucose in that moment, so we have kind of a negative feedback situation where we can then push this over into making glycogen, glycogenesis, instead of making more energy, because we're already getting enough energy out of the lipids. Or all of these things are just intertwined. There's sharing intermediary products and things like that. They're all linked. And this is something that we're going to revisit again in Unit 3, which is called metabolic channeling. And it just means that all of these reactions are interconnected. and they're dependent on each other. And there are things that control whether you're doing one reaction more and another less, or vice versa. There's things that are saying, determining which direction a reaction will be going. So we might be taking glycolysis and pushing it backwards. It can't go backwards. And then you are making glucose out of pyruvate. So all of these things, all of the different regulatory mechanisms that we have can get super complex, which is the point of this, is just to see the actual complexity and to introduce this idea of metabolic channeling, that it's all very determined, it's very controlled. Those reactions are being pushed in different directions by various feedback mechanisms, usually negative feedback, but just some sort of feedback that is determining which way those reactions are going. And we're just going to see examples of this as we go throughout this semester. Okay, now I realize this is a pretty long video. We're getting close to the end, maybe another 10 minutes or so on this video. I think that was the worst of it. So usually the videos won't be this long. This is just longer because it's the introduction video. But bear with me. When we look at this slide is showing the same stuff that we saw here, but not in as much ugly detail. It's nicer to look at. It's easier to look at. So this is a metabolic map that is just more simple. It took all the products and enzymes, or most of the products and enzymes out, and it just put kind of the main point here. But you can see in the middle we've got our glucose situation. They have it labeled carbohydrates. Glucose is a carbohydrate. And we're just talking about how we break down our carbs and get them into, through glycolysis and into pyruvate and acetyl-CoA, which can go into the Krebs cycle, which here they call it the citric acid cycle, which is another name for the Krebs cycle. You can call it whichever one you like. I tend to use the Krebs cycle, just that's how I learned it. It's kind of the old-fashioned way, but either way is completely correct. So anyway, we've got our carbs in the middle here. We have a little bit of glycogenesis going on here to make glycogen. Here we have breaking down the glycogen to get glucose again. So that's the opposite direction, reversing that reaction. So this is all pretty familiar from just the beginning of this video here. We also have, if we take our pyruvates and we don't turn them into acetyl-CoA, say you don't have oxygen, because you need oxygen for the Krebs cycle, those pyruvic acids will just turn into lactic acid. And that's something that happens when you're using energy at a really fast rate, so you can't keep up with your oxygen demands and you end up just fermenting your pyruvates into lactic acid. But this can be reconverted back into pyruvate in the liver. We'll talk about that too eventually as we get through the semester. So there's a whole thing going on here. This is the whole glycolysis to Krebs cycle to electron transport chain and getting energy and all that. And then over here we have our proteins, which are made of obviously amino acids, and we can be breaking them down, putting them into the Krebs cycle. We can be creating proteins, going the other direction, making amino acids or trading amino acids. The urea cycle is something we're going to look at when we get into Unit 3. So we'll see that again. It's just taking essentially the nitrogens and how we deal with the nitrogens from amino acids when we're trying to put them into the Krebs cycle. So we'll deal with that another time. Fats and lipids, that's our Unit 2. We can be breaking them down, making them into energy, getting them into acetyl-CoA, just like we did over here with proteins, or what we did with the carbohydrates. We can also be making fatty acids, making lipids. So here they're calling it lipogenesis. We can call it also fatty acid synthesis. Same thing, so that will be our Unit 2. So here you just see in a nicer kind of way how everything is interconnected going on right here. Some of it happening in the mitochondria, some of it happening out in the cytoplasm. Okay, so that's where we're going here with this slide. Where do these reactions occur? The cytoplasm. The book calls the cytoplasm cytosol a lot. They're synonyms. They mean the same thing. You can call it whichever one you want. I tend to use cytoplasm. That's the thing that the term that I usually use, that I think of when I think of cytoplasm. I think of that word. But cytosol works also. Mitochondria, we all know about the mitochondria. Our next video is going to be even more about mitochondria. We'll talk about mitochondrial structure and what's going on in the mitochondria. But we know that glycolysis happens in the cytoplasm, and Krebs cycle electron transport chain happens in the mitochondria. So we're going to be spending a lot of time talking about the cytoplasm and the mitochondria. Now, some reactions also happen in the endoplasmic reticulum a little bit. We're not going to spend a whole ton of time talking about the endoplasmic reticulum reactions. There's also some reactions, not listed over here, but I have it circled over here, in something called peroxisomes. You may not be able to see that as it's written there. But these are little organelles within the cell that are important for something called reactive oxygen species, or ROS. These may sound familiar to you when you think of free radicals, superoxides, things that just have kind of an unstable amount of oxygen on them. They are damaging to a cell. And they're a normal byproduct of cellular metabolism, but they can also become abnormal if they are too much. So you want to be able to keep those in check, and you do that with antioxidants. And I know everybody's heard of antioxidants before. We're going to spend some time in Unit 2 talking a lot about antioxidants. Super interesting how they work and how they help essentially buffer or detoxify these reactive oxygen species. Really, really good for keeping damage at bay. So we're going to talk a little bit about peroxisomes and reactive oxygen species. Okay, so this is pretty much all we're going to talk about in the endoplasmic reticulum. But in the endoplasmic reticulum, you make proteins, you make lipids, so fatty acid synthesis, and you also store calcium in the endoplasmic reticulum so that you can release it then into the cytoplasm. And calcium is important for a lot of things. You know that calcium is important for all kinds of things within the body. We're going to be really focusing on the calcium at a cellular level in terms of what it's doing in the mitochondria. And what it's doing in the mitochondria is working with enzymes in the mitochondria and helping them to actually work. So calcium is something called a cofactor for these enzymes. So many of the enzymes. And what that means is when something is a cofactor, it is either necessary for the enzyme to work, or it is simply very helpful. It makes the enzyme work better. So it depends on the enzyme, but if you have a cofactor, it's nice to have that cofactor around to help that enzyme to do its job. And the enzyme may not be able to do its job at all if it doesn't have that cofactor. So that is really what's most exciting here about the endoplasmic reticulum, is that it's able to release calcium out into the cytoplasm, where it can then be transported into the mitochondria, and then it can allow those enzymes to work. And in our next video, we will see calcium moving through various transport mechanisms, getting into the mitochondria, and how that can help these enzymes to work. Okay, now in the cytoplasm, we have glycolysis, we know that. We also have fatty acid synthesis, so lipogenesis is happening out in the cytoplasm. The pentose phosphate pathway, we mentioned this was that shunt from glycolysis. The pentose phosphate pathway makes the sugar component of DNA and RNA. So ribose and deoxyribose. You don't have to memorize those words. I didn't even need to say them, but just the sugars for DNA and RNA. And it also makes not just NAD, not just NADH, but NADHP. That's just NADHP or NADH with a phosphate added. So this is a high energy compound. And we're going to look at the pentose phosphate pathway in more detail later on in Unit 1, so in the coming weeks. So we'll see a bit of that and what we use this NADP. That's a tongue twister. NADHP. Okay, now in the mitochondria, we have the Krebs cycle, or the citric acid cycle, whichever one you want to call it. Beta oxidation, that's where we break down fats and turn them into acetyl-CoA, so we can run that through the Krebs cycle. Ketogenesis, and the urea synthesis. So I'll mention what those are in a second, but I want to just say that the ketogenesis and urea synthesis is predominantly happening in hepatocytes. Hepatocytes are liver cells, so these liver cells are really good at doing a lot of creating nutrients or breaking down nutrients, as well as detoxifying all kinds of things. So that's what hepatocytes' purpose is. So they're essentially breaking down proteins, and you get these ketone bodies, and urea, and also ammonia as a byproduct of breaking down proteins. Now, the ketone bodies, so it's written here, it got all, I wrote notes on it earlier so you can't see, so just pretend you can't see that. I'll rewrite all that. So with ketogenesis, we're making the ketone bodies, and these ketone bodies are coming from either proteins, usually proteins, but also from fats. So you go, okay, well, if you're getting urea and ammonia, these two are not good things to have. You know, you don't like these things. These are toxic. So why do you want to make these ketone bodies if this is what you're going to get in addition to them? But these ketone bodies are good for energy for the brain in the fasting state. So your brain cells can only use glucose or ketone bodies for energy. They can't do beta oxidation. They can't take a fat and break it down into energy in the same way that a typical cell could. They need these ketone bodies or glucose in order to have energy. So when you don't have glucose in your blood, so when you're hypoglycemic, so that means low glucose in your blood, then you gotta have these ketone bodies. So you gotta do ketogenesis. Now, this fasting state, you probably think like, okay, well, that's when people do fasting. You know, they don't eat for a day or several days or whatever, however long they're deciding to do the fasting for. But that's not what it means metabolically exactly. The fasting state is simply talking about when you haven't eaten for a few hours. So a typical meal, it runs through in about 3 to 4 hours-ish, maybe 5, depending on how heavy the meal was. So if you say eat your dinner at 6 o'clock, and then whenever you get up and get going and have your breakfast, it's probably been at least 12, 13, 14, maybe 15 hours until you have breakfast. That's a fasting state. So you've become hypoglycemic while you're sleeping. Your body started breaking down proteins and fats, and you're getting ketone bodies, and your brain is happy about that. But then you got to deal with these toxic byproducts and get rid of them. You also have to deal with the possibility of ketogenesis. That's not that. Ketoacidosis, sorry. exhausting myself with my own video, so I can't even say the right word. Ketoacidosis is something that happens if you have too many ketone bodies. So if the fasting were to continue for too long, then you can end up with ketoacidosis, and that is not a good thing. You don't want to have your pH balance thrown off and be in an acidic state. So ketone bodies good, urea, ammonia not so good, ketoacidosis bad, right? Okay, so that does it for our first video. Our next video will be all about mitochondria and what's going on in the mitochondria and all the different structures in the mitochondria.